Hi everyone. In this video I am going to explain you about eukaryotic cell structure. Under this, cell wall and cell membrane are plasma membrane.
Eukaryotic cells. The eukaryotes include all the protists, plants, animals and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through presence of membrane-bound cell organelles. Eukaryotic cells possesses an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All protists, plants, animals and fungi are the best examples for eukaryotic cells. Eukaryotic cell shows extensive compartmentalization of cytoplasm because of presence of membrane-bound cell organelles. Eukaryotic cells possesses well-organized nuclear membrane. In addition, eukaryotic cells have variety of locomotory and cytoskeletal structures. Genetic material in eukaryotes organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell walls, plastids and a large vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all higher plants. Let us now look at individual cell organelles to understand their structures and functions. Remember, all eukaryotic cells are not similar. For example, plant and animal cells are eukaryotic cells, but they shows differences in different cell organelles. Cell wall, plastids and large central vacuole present in plant cell, but these are absent in animal cells. On the other hand, animal cells have centrioles which are absent in plant cells. Let us now discuss about each and every individual cell organelle in eukaryotic cells. The very first one, cell wall, a non-living rigid structure called the cell wall. Cell wall forms an outer covering for the plasma membrane of bacteria, some protistans, fungi, algae and plants. Algae have cell wall made up of cellulose, galactans, mannans and minerals like calcium carbonate. In other plants, cell wall consists of cellulose, hemicellulose, pectins and proteins. Fungal cell wall is made up of chitin. Cell wall is the outer covering of plasma membrane in bacteria, some protistans, fungi, algae and plants. We already discussed cell wall is absent in animal cells. Algal cell wall is made up of cellulose, galactans, mannans and minerals like calcium carbonate. Plant cell wall made up of cellulose, hemicellulose, pectins and proteins. The fungal cell walls are made up of chitin. Bacterial cell wall is made up of peptidoglycon. See, cell wall of plant cell. Cell wall of plant cell made up of four regions: primary wall, secondary wall, tertiary wall. In addition to that, middle lamella. Primary wall is thin and elastic, composed of cellulose, hemicellulose and pectin. Gradually diminishes as cell matures, capable of growth. Secondary wall, rigid, thick, composed of cellulose, hemicellulose, pectin, absent in meristematic cells. Inner side of primary wall present towards cell membrane. Tertiary wall present only in tracheids of gymnosperms, composed of hemicellulose and xylem. Middle lamella, common layer between two cells. Let's see simple flow diagram of plant cell wall, which is made up of primary wall, secondary wall, tertiary wall and middle lamellum. Middle lamella is consist of calcium and magnesium pectates, which are called plant cement. Amount of calcium is more than magnesium. Calcium is main constituent of cell wall, but addition to cellulose, hemicellulose, cutin, pectin, lignin, suberin are also present in cell walls. Cell wall worked as frame or protective layer of cell or exoskeleton of cell. Cellulosic microfibrils and macrofibrils are arranged in a layers to form skeleton of cell wall. In between these layers, other substances like pectin, hemicellulose may be present. These form matrix of cell wall. Middle lamella composed of calcium pectate and magnesium pectate. In that also calcium is more. Cellulose is main constituent of cell wall of plant cell, and they also contains hemicellulose, cutin, pectin, lignin, suberin. Cell wall is the protective layer of cell, which is also called exoskeleton of cell. Cell wall contains cellulosic microfibrils and macrofibrils. In between these microfibrils and macrofibrils, they contains hemicellulose and pectin. So these cellulosic microfibrils and macrofibrils forms skeleton of cell wall. Let's see the diagrammatic representation of cell wall of plant cell. The outermost one is primary cell wall, inner to that secondary cell wall. Secondary cell wall is made up of secondary wall 1, secondary wall 2, secondary wall 3. Among these, secondary wall 2 or S2 is thickest. Cell wall materials like hemicellulose, pectin, lignin are synthesized in plant Golgi bodies or dictyosomes. Formation of cellulose occurs by cell membrane. Plasmodesmata. Plasmodesmata name proposed by the scientist Strasburger in 1901. These are cytoplasmic connections between two adjacent plant cells. Plasmodesmata are characteristic of multicellular plants. Endoplasmic reticulum tubules, which are called desmotubules, helps to maintain continuity of cytoplasm. The cell wall and middle lamella may be transversed by plasmodesmata, which connect cytoplasm of neighboring cells. Cell wall materials like hemicellulose, pectin, lignin synthesized from Golgi bodies or dictyosomes. Cellulose formed from cell membrane. Plasmodesmata name given by Strasburger. Plasmodesmata are cytoplasmic connections between two adjacent plant cells. Generally, endoplasmic reticulum tubules or desmotubules helps to maintain continuity of cytoplasm between adjacent cells. The cell wall and middle lamella may be traversed by plasmodesmata, which connect cytoplasm of neighboring cells. Functions of cell wall. Cell wall gives shape to the cell. Cell wall protects the cell from mechanical damage and infections. Cell wall also helps in cell to cell interaction. Cell wall provides barrier to undesirable macromolecules. Cell wall gives shape to the cell. It protects the cell from mechanical damage and infection. It also helps in cell to cell interaction, and cell wall provides barrier to undesirable macromolecules. The next one, cell membrane or biomembrane or plasma membrane. Biomembranes are cell membrane or plasma membrane. The biochemical investigation clearly revealed that the cell membranes possesses lipid, protein and carbohydrate. The ratio of protein and lipid varies in different cell types. In human beings, the membranes of the erythrocyte or RBC has approximately 52% protein and 40% lipids. It is universal. Structure of cell membrane of prokaryotes is similar to the eukaryotes. Average composition of cell membrane. Lipids 40%, In those, phospholipid, cholesterol, glycolipids. Proteins 58 to 59%, majorly composed with the amino acids arginine and lysine. The biochemical investigations revealed cell membrane possesses lipid, protein and carbohydrate. The ratio of protein lipid varies in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% protein, 40% lipid. Cell membrane is universal. Structure of cell membrane of prokaryotes almost similar to eukaryotes. Average composition of cell membrane. Average composition of cell membrane contains 40% lipids, 58 to 59% proteins, 1 to 2% carbohydrates. Structure of biomembranes. Under this, the very first one, sandwich or trilamellar model, which is introduced by the scientists Davison and Danieli in the year 1935. According to this model, the plasma membrane is made up of three layers in which a bimolecular layer of lipid is sandwiched between two single layers of proteins. According to this model, each protein layer is 20 angstroms thick and bilayer of phospholipid is 35 angstroms thick. Thus, total thickness is 75 angstroms. This model is also called PLLP model, which means protein, lipid, protein structure, range 75 to 100 angstroms. Phospholipid molecule called as amphipathetic molecule due to presence of two types of parts, hydrophilic head and hydrophobic tail. Sandwich or trilamellar model introduced by Davison and Danieli. According to this model, lipid bilayer covered by proteins on both the sides. According to this, the thickness of this membrane is 75 angstroms. And phospholipid molecule called as amphipathetic molecule due to having both hydrophilic heads and hydrophobic tails. Hydrophilic head of the phospholipid binds with protein layer by hydrogen and ionic bonds. Hydrophobic tail of phospholipid are attached to each other by van der Waals forces. Let's see the diagrammatic representation of sandwich model or PLLP model, protein lipid, lipid protein model. See, proteins are present both the sides of phospholipid bilayer. Let's check this diagram carefully for your better understanding. Unit membrane model, introduced by the scientist Robertson in the year 1959. According to unit membrane model, all the cellular and organellar membranes are structurally and functionally similar. Difference in chemically and size, but structurally and functionally similar. Both of the above models are rejected because they fails to explain the selective permeability of plasma membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, the chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. Third one, fluid mosaic model introduced by the scientists Singer and Nicholson. This is last and most widely accepted model for the structure of plasma lemma or plasma membrane or cell membrane or biomembrane. According to fluid mosaic model, proteins are arranged in phospholipid bilayer as mosaic pattern. Thus, membrane is termed as protein iceberg in a sea of phospholipid or gulab jamun protein in a concentrated solution phospholipid of sugar. Phospholipids. Phospholipid is the main component of cell membrane because it forms continuous structural frame of cell membrane. The study showed that the cell membrane is composed of lipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar heads towards the outer side and the hydrophobic tails towards the inner part. This ensures that the nonpolar tail of saturated and unsaturated hydrocarbons is protected from the aqueous environment. The lipid component of the membrane mainly consists of phosphoglycerides or phospholipids. Phospholipid layer provides fluidity to plasma membrane. Because phospholipids are rich in unsaturated fatty acid which are liquid in nature. The quasi-fluid nature of lipid enable lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important in various functions like cell growth, formation of intercellular junctions, endocytosis, secretion, and cell division. Phospholipid bilayer provides fluidity of plasma membrane. This quasi-fluid nature of lipid enable lateral movement of protein within the overall bilayer. The fluid nature of membrane is also important in various functions like cell growth, intercellular junction formation, endocytosis, secretion, and cell division. Cholesterol is also present in plasma membrane. Cholesterol are more rigid than phospholipid, so it helps in stability of membrane structure. Cholesterol is absent in membrane of prokaryote, thus haploids. Pentacyclic sterol provides stability to prokaryotic cell membrane. Cholesterol also present in plasma membrane, which is more rigid than phospholipid, which helps in the stability of membrane structure. In case of prokaryotes, cholesterol is absent. Instead of that, they contains haploids, pentacyclic sterol, which gives stability to prokaryotic cell membrane. Proteins. Two types of proteins are present in plasma membrane on the basis of ease of extraction. The very first one, integral or intrinsic proteins. These proteins are tightly binds with phospholipid, thus they cannot easily removed from membrane. Integral proteins are of two types, partially buried, totally buried. Some integral proteins which are totally buried through the complete thickness of membrane. These type of proteins are called as tunnel or channel proteins, which provide a passage for movement of water soluble materials across the membrane. Second one, peripheral or extrinsic proteins. These are superficially arranged on the surface of lipid bilayer and can be separated easily. These proteins have enzymatic activity. Oligosaccharides, sugars of the glycolipids and glycoproteins on the outer surface of plasma membranes are involved in cell-to-cell recognition mechanism. The best example of cell recognition is fertilization, where sperm and egg recognizes to each other, and blood antigens. Let's see the diagrammatic representation of fluid mosaic model of plasma membrane. See how lipid bilayers are arranged, how integral proteins and peripheral proteins are arranged, how sugar molecules are present in cell membrane, how cholesterol present in lipid bilayer. Let's see another diagram of fluid mosaic model of cell membrane for your easy understanding. In lipid bilayer, how hydrophilic heads present towards outer sides, hydrophobic tails present towards inner sides. Integral proteins, peripheral proteins, sugar molecules, and everything. Let's check these diagrams carefully for your better understanding. Transport through plasma membrane. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without requirement of energy, and this is called passive transport. Neutral solute may move across the membrane by the process of simple diffusion along with concentration gradient, which means from higher concentration to the lower concentration. The major function of plasma membrane is transportation of molecules across it. Plasma membrane performs both passive transport and active transport. The transportation without requirement of energy is called passive transport. The transportation occurred with the help of energy in the form of ATP called active transport. Neutral solutes may move across the membrane by the process of simple diffusion along with concentration gradient, which means from higher concentration region to lower concentration region. Water may also move across this membrane from higher to lower concentrations. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, which means from lower to higher concentrations. Such type of transport is an energy dependent process in which ATP is utilized and is called active transport. Example, sodium pump, potassium pump. Plasma membrane also performs water movement across the membrane from higher to lower concentrations called osmosis, and also performs active transport with the help of energy in the form of ATP called active transport. Example, sodium ion, potassium ion pumps. Endocytosis or bulk transport. It is two types. Pinocytosis or cell drinking. Ingestion of liquid material by plasma lemma in the form of vesicles or bag-like structures, pinosomes, is called pinocytosis. Phagocytosis or cell eating. Ingestion of solid complex materials by membranes in the form of vesicles, phagosomes, is called phagocytosis. Second one, exocytosis or immunocytosis or cell vomiting. Ejection of waste materials from cell through plasma membrane is called exocytosis. Endocytosis classified into two types, pinocytosis and phagocytosis. Pinocytosis is called cell drinking, phagocytosis is called cell eating. Exocytosis, which is also called amniocytosis or cell vomiting. Key points. Plasma membrane is an asymmetrical structure. The lipid component of the membrane mainly consists of phosphoglycerides or phospholipids. One of the most important functions of plasma membrane is the transport of molecules across it. Unlike animal cells, every plant cell is surrounded by a polysaccharide cell wall. So, plasmodesmata enables direct, regulated intercellular transport of substances between neighboring or adjacent plant cells. In the cell membrane, non-polar tail of saturated and unsaturated hydrocarbons is protected from the aqueous environment. You have to remember all these key points. Up to here, eukaryotic cell structure. Under this, cell wall and cell membrane or plasma membrane is completed. In next video, I will explain you about endomembrane system, endoplasmic reticulum, Golgi bodies, lysosomes and vacuole. Thank you very much.